Discuss the spermatogenesis. Now, spermatogenesis is the process by which the gametes or germ cells are produced from the germinal cells of the gonads. So, these primordial germ cells are basically the initial cells which have to form the functional gametes during embryonic development. So, these primordial germ cells they arise at different sites from that of gonads, and once the gonad develop, they migrate into them and become established. Hence, these cells have an extra gonadal origin in most invertebrates and vertebrates. In sexually reproducing animals, there are two kinds of gametes, that is female gamete and male gamete. The female gamete or ovum or egg is invariably larger, non-motile and nutrient-filled cells, and male gamete or spermatozoon or sperm is active, mobile and varies greatly in size and constitution. The formation of female gamete, ova or egg is known as oogenesis, and the formation of the male gamete or sperm is termed as spermatogenesis. Now, what is this spermatogenesis? It is a process by which the spermatogonia are transformed into sperms. So, in the different stages which are occurring during spermatogenesis are spermatocytosis, meiosis, and spermiogenesis. And this whole process is taking approximately 64 days in humans, and this process is occurring in the seminiferous tubules of the testes. Spermatogenesis begins at puberty and continues to old age. At birth, the primordial germ cells in male infant are large cells which are surrounded by the sporting cells called as tentacular cells or Sertoli cells. Shortly before puberty, the sex cords acquire a lumen and become seminiferous tubules. Then primordial germ cells give rise to spermatogonial stem cells. Now these are the sequences of spermatogenesis. Now this spermatogonia which are arising from the primordial germ cells, so these are of two types, that is type A and type B with 44 plus XY number of chromosome. Now type B spermatogonia they enlarges to form the primary spermatocyte, and this process of conversion of spermatogonia to spermatocyte is known as spermatocytosis, which takes 16 days. Now after that, the primary spermatocyte undergo first meiotic division, which is the reductional division, and there is a haploid number of chromosome they are formed. So two secondary spermatocytes they are formed. Now this primary spermatocyte production, then secondary spermatocyte, the second meiotic division it happens, which is not the reductional one, so they form the spermatid. So these four spermatid are formed. Now starting from this primary spermatocyte to the spermatid, this period is known as meiosis, which takes around 24 days. Now this spermatid they undergo metamorphosis to form the spermatozoa, and this process is known as spermiogenesis. It takes 24 days, and after that the spermatozoa, the capacitation, it happens. Now what is this spermiogenesis? It is the process of metamorphosis from spermatid to spermatozoon. You can see the sequence of events. This diagram is taken from your standard textbook. Now the nucleus condenses to form the sperm head. Golgi apparatus forms acrosomal cap. Centriole, now here the centriole are present. They divide into proximal and distal, and proximal centriole it is lying in the neck region. Here the distal centriole become a ring-shaped structure called as annulus. Mitochondria, it forms a spiral sheath around the axial filament in middle piece. Remaining part of the axial filament, it forms the tail, and most of the cytoplasm of the spermatid is shed, but the cell membrane persists as a covering for the spermatozoon. Now finally spermatozoa, they are released in the lumen of the seminiferous tubule, and this process is known as spermiation. Now what is capacitation of spermatozoa? Process of conversion of immature spermatozoa to mature spermatozoa. So it is occurring in the female reproductive tract during the fertilization. Before the fertilization, the uterus or uterine tube is the site where this capacitation of spermatozoa it happens. Now this process it involves the removal of the acrosomal cap, you can see here, alteration of the glycoprotein coat over head of sperms, and removal of seminal proteins from the head of sperm. The effect of this capacitation is it increases the sperm mobility, destabilizes acrosomal membrane, and allows to penetrate outer layer of ovum. Now this is the structure of sperm. The sperm has head, neck, middle piece and tail. You can see head, neck, middle piece and tail. Head is covered by acrosomic cap or gallia capitis. Neck is narrow, containing basal body and centriole. Axial filament begins just behind the centriole and passes through the middle piece and extends into tail. At the part where the middle piece is joining the tail, the axial filament, they pass through a ring-like structure called as annulus. The part of the axial filament lying in the middle piece is surrounded by the mitochondrial sheath. So their spiral sheath made up of mitochondria is there in middle piece. Now, what are the abnormalities of sperm? This is a normal sperm. This is a sperm with small head, two sperm heads with common tail, bifurcated tail and long tail. So this is all about the spermatogenesis. Thank you very much.